Good morning, and thank you all for joining today's webinar, We Dig Bio, Spotlight on Herbarium Research with the New York Botanical Garden. My name is Charles Zimmerman, and I am so pleased to welcome everyone who's joined today to learn about biodiversity collections and plant science during We Dig Bio 2021. At any time during today's event, please feel welcome to submit questions by clicking the Q&A button at the bottom of your window, and our host will raise these for our speakers to answer at the end. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. This presentation is being recorded, and it will be shared for future viewers on the NYBG Lecture Library. Worldwide Engagement for Digitizing Biocollections, or We Dig Bio, is a semi-annual 4-day event led by museums, universities, herbaria, and other organizations working to preserve and share natural history specimens with researchers and the public. Our common aim is to mobilize community scientists to help create digital data for biodiversity collections and support their extended use for understanding nature. Over the next 3 days, thousands of individuals around the world will join together for virtual workshops, collections tours, research talks, and citizen science expeditions. You can learn all about the great events happening by checking out WeDigBio.org or follow WeDigBio on Twitter. This morning's webinar marks the third in an exciting series of We Dig Bio events hosted by the New York Botanical Garden. We began yesterday with a historical perspective of NYBG's scientific collections with our virtual herbarium tour. We then dove deep into the process of participating in New York Botanical Garden virtual expeditions for a citizen science workshop. Today, I'm thrilled to welcome 3 contemporary plant scientists to share perspectives on how herbarium collections and their digitized data enable modern biodiversity research. Our first speaker, Dr. Matthew Pace, is Assistant Curator of the William and Linda Steer Herbarium at NYBG. His research interests focus on orchids, their evolution, relationships, morphology, taxonomy, and biogeography. As principal investigator for the NSF-funded Digitizing Endless Forms, facilitating research on imperiled plants with extreme morphologies, Matthew coordinates efforts between 17 US herbaria and hundreds of citizen scientists around the world to document botanical collections and aid researchers seeking to study and conserve plant biodiversity. Please join me in welcoming Matthew Pace.
Thank you for the introduction, Charlie, and thank you to everyone for attending. Today, I'm going to share a bit about how herbarium specimens and digital data help inform and drive my research. I work on orchids. Other than their obvious beauty, orchids are a great family in which to do research because they are so diverse and they often face acute conservation concerns. Orchids are among the 3 largest plant families, with over 28,000 known species and more described every single year. Because of this diversity, we can ask really interesting evolutionary and ecological questions, like why are there so many species of orchids? Why are orchids particularly diverse in the tropics, and why are so many orchids epiphytic? And why are so many orchids extremely rare? And what conservation protocols can we develop to protect them? For many of these big questions, herbarium specimens and digital data provide a critical resource from which we can try and answer these questions. So how exactly do I study orchids? Although I use a diversity of tools from fieldwork to molecular analyses, herbarium specimens and digital data inform every part of my research. Specimens and digital data are incredibly powerful because they allow me to study a species across its entire distribution. This allows me to understand differences between populations across space. It would take me years to visit all of the herbaria across the US, But by digitizing these specimens, this allows me to view hundreds or even thousands of specimens at one time. Secondly, herbarium specimens are unique because they allow me to also view a species through time, as specimens have been collected in an unbroken chain for the last 300 years. This allows me to understand how species and populations may be changing over time. Equally important to my research is how you help me study orchids. Most importantly, your support of the US National Science Foundation allows NSF to provide grants to institutions to digitize their collections. NYBG is the lead institution on such a grant, as Charlie referred to, and this is called Digitizing Endless Forms, which digitizes specimens of epiphytic, succulent, and carnivorous plants. A partnership among 17 museums, botanical gardens, and universities, this grant has digitized about 2 million specimens, including 400,000 orchid specimens. The name for this grant comes from a line in Charles Darwin's Origin of Species. Quote, "There is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved." And so the purpose of this digitization grant is to help us understand this diversity. After specimens are digitized here at your botanical garden or one of our other partner institutions, citizen scientists like you help us transcribe the data on the specimen label. After it's transcribed, these data are sent to data aggregators like iDigBio, the NSF-supported National Digital Collection, where they can be used by anyone, from researchers to high school students. Now, I want to focus on a project of mine that leveraged these digital data to understand potential population and distributional changes of orchids in New York State. So across North America, different areas have different species that primarily occur in that particular area. For example, northern species occur across Canada and Alaska and dip down into the northern part of the United States, whereas there's basically a separate suite of species that only occur in the southern part of the United States. And New York is really interesting because it occurs right at the juncture point of these two biogeographic regions. And so if you're interested in questions about how populations may be changing over time due to climate change or the overpopulation of deer or pollution from industrialization, New York is a really interesting place to study how these two groups of species may be responding. And so to try and look at this question, I identified 10 different species of orchids, 8 that are primarily northern in distribution and reach one of their southern limits in New York State, and 2 that are primarily southern in distribution and reach their northern limit in New York State. First, I verified the identity of all the specimens and their locations, looking for outliers and just making sure the data were clean. Then I downloaded the transcribed specimen data from data aggregators like iDigBio. Based on the collection year that's transcribed from the label, I divided specimens into multi-decade categories. For example, all specimens collected from 1950 to 1999 were put in one particular category. And I did this for all the specimens across all the species. And then I worked with NYBG's Geographic Information System lab manager, Liz Geely, to map these specimens and start to understand what patterns are going on here. So the first species I want to talk about is Platanthera hookeri. This is one of the northern species that reaches its southern limit in New York State. It's a really interesting orchid, has big round leaves, and it primarily occurs in coniferous forests that are relatively undisturbed. And if we look at its distribution originally, it was widely found across New York State and actually somewhat common. But unfortunately, as you travel through time, moving closer to the present, we find massive population declines, such that the species is now only found in the northern part of the state in the Adirondacks. Similarly, looking at another northern species that reaches its southern limit in New York, Arethusa bulbosa, or the dragonmouth orchid, which is one of the most beautiful orchids in North America, we again see massive population declines, particularly on Long Island. And that's an area, I'm actually from Long Island originally, and I can tell you there are very few areas where Arethusa bulbosa can live anymore. The species primarily occurs in sphagnum bogs or wetlands, and unfortunately those areas have been drained and developed for housing, such that now there's only one known population on Long Island and a few in northern New York. Moving to two southern species that reach their northern limit in New York, the first is Tipularia discolor. And this species has always been rare, according to herbarium specimens within New York. It was only found in the northern part of Manhattan, as well as eastern Long Island. And unfortunately, this species has also declined. Now, for this species, I'm actually not particularly surprised. Tipularia only occurs in primarily undisturbed forests. And anyone who's ever been to New York or Long Island can tell you there are very few undisturbed forests across Long Island. However, looking at the other southern species that I occurred in this study, I was fairly alarmed. So this species is Spiranthes vernalis, and again, it's primarily southern species and was once widely found across Long Island. However, it too has also undergone massive population declines. And the thing that's interesting about Spiranthes vernalis is that it's actually somewhat of a weedy species. It's native, but it's found along highway medians in Virginia and Delaware and Maryland. It's actually a lawn weed across much of the South. And again, growing up on Long Island, I can tell you there are a lot of lawns on Long Island. So why has this species been under decline? And in fact, why is it not moved northwards, perhaps into the Hudson Valley? These are questions that we're left with that are the next step in this project. And in fact, looking across all 10 species that I included in this study, all of the northern orchids are retreating northwards. However, the southern orchids are also declining. And the specimen data that I gathered indicate that New York State orchid diversity is hollowing out as these northern species are not replaced by their southern counterparts. And so I really want to stress here that herbarium specimens and their digital data are critically important because they allow us to identify these problems and ultimately work towards solutions. You can't fix a problem if you don't know it's a problem in the first place. For example, we can start to ask questions about what is causing the declines of both northern and southern New York orchid species. And here it's likely to be a mixture of pressures that are working synergistically. Perhaps climate change and an overabundance of deer and unchecked development and draining of wetlands are all working together in some way to cause these declines. And these are questions that, again, need more data in order to be able to really answer. And that's something that I always stress when I talk about herbarium specimens, is that the only way we're able to do this type of research is because we have this unbroken chain of specimens across space and time. And I know this is maybe a little bit of a downer talk, so I just want to end on a brighter note. By continuing to collect specimens and transcribe their data, this ultimately leads to new discoveries as well as solutions for problems. So just within the last 5 years, I've been able to describe several new North American orchid species based on herbarium specimens and their digital data. So again, herbarium specimens and data provide the solution to problems. And so thank you to everyone for listening, and thank you to Charlie for hosting this webinar. Thank you.
Terrific, Matthew. Thank you so much for that presentation. We really appreciate it. We are going to be doing questions at the end following all 3 talks today. So audience members, please feel free in the meantime to submit your questions by clicking the Q&A button at the bottom of your screen. Our next speaker Dr. Alejandra Vasco studies the biology of ferns, encompassing their evolutionary history, systematics, taxonomy, and evolutionary development. Her main research interests are the taxonomy and systematics of the fern genus Elaphoglossum and the development and evolution of leaves in ferns. Alejandra obtained her Bachelor's of Science degree from the University of Antigua in her native Colombia, and her PhD as part of the joint program between the City University of New York and the New York Botanical Garden. Currently, she works at the Botanical Research Institute of Texas. Join me, please, in welcoming Alejandra. You're muted, Alejandra.
Okay, I'm sorry. I hope you can hear me now.
Yes, we can hear you. Thank you.
Perfect. So good morning, everyone, and thanks so much to the New York Botanical Garden for this invitation. I'm very happy to be here today with all of you, even virtually, and mostly digitizers and other people interested in natural history collections. I'm also very excited to be talking about some of my favorite topics today with you, which are the fern genus Elaphoglossum and how I use natural history collections, especially herbarium specimens in my research. But before I begin, I would like to mention and acknowledge this other big digitization project, which is the Pteridophyte Collection Consortium, which is also another big digitization project as the Endless Form that Matthew just mentioned is funded through NSF with the aim of digitizing ferns, lycophytes, and their extinct free sporing relatives from 36 herbaria and museums in the US. So this is the page of the Pteridophyte Collection Consortium, and at the bottom you can see also the page of where all the collections, the digitized collections, are available. So I'm very thankful, and I will be talking about digitizing collections, why they are important. And most of the time now I'm using the Teredo portal to get those collections. So thanks so much also all of you, as Matthew said, that support NSF. To begin, I would like to situate you in the branch of the tree of life you'll be hearing about today, the ferns. You can see them here, a lineage of vascular plants that is sister or closely related to seed plants. And within this group, I want to introduce you the main characters of today's talk, which are the species of this amazing fern genus that is called Elaphoglossum. It's not super fern-like because, as you can see, it has simple entire leaves. The genus Elaphoglossum has been my academic partner for over 16 years. And today I want to talk and tell you two little stories about it. So as I just told you, Elaphoglossum is very easy to recognize from other fern genera because of its simple entire leaves. So you see, they just look like a tongue. And also because fertile and sterile leaves are dimorphic, meaning they have two different shapes or morphology. Most of the species of Elaphoglossum are epiphytes. So this is perfect because we have a fern that it's an epiphyte, so perfect for this symposium. And epiphytes also just means that the ferns live over the trees. They don't get inside any tissue, so in that way they don't damage the trees. They just use them as a substrate to live there. Elaphoglossum is one of the most diverse genera of ferns worldwide, with around 600 species. And this is a map I took from the Teredo portal again, and these are all the digitized collections that we have available. And this is actually a very, very nice proxy of the real distribution of Elaphoglossum. So between these two lines that you see here is where most species of Elaphoglossum occur. So the dots that you see outside of these two lines are probably collections that need to be corrected for their coordinates. And this is something I wanted to also thank digitizers and just mention here that digitized collections are a work in progress always. There's always more things to do, things to correct, more collections to digitize. So it's not that when we digitize everything that we have, the work is gonna be over. We need to really keep supporting in many different ways these collections and these digitized collections so that they can be relevant for a long time. So coming back to Elaphoglossum, you can see that it's very diverse all over the tropics, and also the density of the dots that you see here reflect very well the different centers of diversity that Elaphoglossum has. So you have a big center of diversity in the neotropics, where 70% of the species of Elaphoglossum occur, but we also have two diversity centers in Madagascar and in Southeast Asia, with the other, where the other 25% of the species of Elaphoglossum occur. So this is a cartoon or a phylogeny, a tree of life, with a representation of the main clades that we have from Elaphoglossum. We have 7 main clades, or 7 main groups of branches. And I just wanted to point out that in Elaphoglossum, again, 99% of the species have simple leaves. And here you can see more examples of this diversity of this fern, epiphytic mainly fern genus, with simple leaves. So I just say 99%. So what happened with the other 6 or the 1% of those species? Well, yes, they have divided leaves. They are very rare in Elaphoglossum, but they are 6 species, and they occur in 3 different groups, where you can see here the arrows. These are the 3 different groups where species of divided leaves of Elaphoglossum occur. But there is one group, Elaphoglossum, this Squamipedia, the section Squamipedia, that has 4 species. So 4 of the species, of the 6 species of Elaphoglossum that have divided leaves, occur in this clade that you are going to see that it's not very big. So that was incredible for me, that studying Elaphoglossum, always simple leaves, to see that in this group that is very little, we have all this diversity of divided leaves. So we wanted to study a little bit more, and we did a lot of things. I'm going to present here 2 of the questions we have. So we really wanted to know how many species have divided leaves in section Squamipedia, and how many times divided leaves have evolved. And you are going to see that we use a lot of collections and a lot of digitized collections to be able to answer these questions. Let me show you a little bit of the diversity. These are live plants, and these are how these little plants of section Squamipedia look like. They are no more than 15 centimeters, so they are pretty cute terrarium plants, but they grow in the neotropics, right? So they don't need terrariums to survive there. Just for fun, I wanted to show you the diversity in herbarium specimens. These are the same species here and here, but you can see how different they look, but how important is to have this natural history collections. I always say that having natural history collection is like having a photograph in time of that species, and it gives you a lot of information. Matthew already talked about what we can do sometimes with collections, just to see how the ranges have, the distributional ranges have changed through time, but you can also see a lot of characters, morphological characters, that allow you to distinguish species. So to answer the first question about how many species with divided leaves were part of the section Squamipedia, we did a taxonomic treatment. And a taxonomic treatment is just a comprehensive treatment of a taxon, when you review all the known species, all the literature within a group, and you also describe new species. And so you synthesize all this available information in a piece of work, when you just discuss the species, talk about geographic distribution, morphological variation, etc. So after doing all of this, what we did was we review collections from 30 herbaria. We physically observed 3,000 herbarium sheets, and that was also very important. So sometimes digitized collections are very important to know where things are. So taxonomists many times do need to look at the specimen itself, but the digitized collection lets you know where they are, where do you have to go, or which whose herbarium you have to visit in order to see this collection. So over 3,000 herbarium sheets, and we did field work in Colombia, Costa Rica, Ecuador, and Brazil. So with that, we recognized 16 species in this clade, 3 new species. We described 3 new species to science, all of them with simple leaves. And then finally, we decided that there were 4 species with divided leaves in this group. So to answer the second question of how many times divided leaves have evolved in this clade, we use phylogenetic approaches. So this is just kind of like knowing your tree of life. So we also reconstruct the tree of life of these species. And what we found, you can see here that the blue squares are the ones that are divided species. All of the other species have simple leaves. So what we found doing this phylogeny using molecules, and many of the specimens we have here were also from herbarium collections, we found that divided leaves evolved independently 4 times within the group from ancestors with simple entire leaves. But that, yeah, so it was 4 different times that these divided leaves evolve within this group. Okay, so we have these 2 questions answered. How many species? 4 of divided leaves, and how many times? Also 4 times independently. And so I already mentioned that to be able to do this, we use and made many herbarium collections. So let me tell you 2 specific examples within this project of how digitized collections and collections in general impacted this research. And well, the last example is very, very close to my heart, so you're gonna see why. So here I'm just telling you, and this is probably something you have thought about many times, but I just wanted to point it out, that using digitized collections, herbarium collections, you can travel. Because as I told you, I did field work in these 4 parts of the world when I was doing this taxonomic treatment and this phylogeny. I did Costa Rica, Colombia, Ecuador, and Brazil. But if you see the distribution of the whole of the old species of this section, you see there are many more dots in many more countries. But it gets expensive, and it gets also difficult sometimes to travel and collect. But these natural history collections take us to those places we can observe, and we can put dots in this map to be able to know really what the distributions of the species are. So that's why I say that using digitizing herbarium collections, you can travel. But also, and this is the example that is close to my heart, using digitized herbarium collections, we were able to rediscover species. So these were the pinnate species. So they are the most fern-like Elaphoglossums that you can see, because the divided leaves are pinnate. And these are not very well collected. We have probably digitized around 107 digitized collections of these ferns. And these include duplicates. So it might be less collections. I think I went to Colombia and I looked for these species a lot. I only did 2 collections, like in 4 or 5 trips. And my hypothesis is that this species grow in the canopy up in the forest. So unless you just go and climb that tree, you are not going to find it. So I think the collections that are available is because branches fell to the ground and we were able to collect them. So it was very important to use collections to really understand what was going on with this species. So when we started to observe all these collections available, what we saw is that there were actually 2 forms. One form that has less pinnate, the one in the upper part of the slide, that it has less pinnae and the pinnae at the base were wider. The other ones at the bottom have much more pinnae, and the pinnae, these segments, were really thin at the base. So we were already seeing some patterns. And when we plot them in the maps, we saw that the ones with less pinnae are just restricted to the western part of Colombia, the country. This is South America. And then the ones with more pinnae are actually only restricted to Peru and Bolivia. Those were two countries that I never did field work for this. I actually have never done field work there, so I wish I can go hopefully soon. So we saw that there are two different entities, and they had names. So one was Elaphoglossum columbianum, and the other one's Elaphoglossum morii. What happened was that because of lack of knowledge, and also because there were not many digitized collections, all these collections were put in morii. But we know now that there are these two different species. And I was saying that this is very close to my heart because I'm from Colombia, native from Colombia, as Charlie said in the introduction. And I was really happy to be able to kind of like take out that species and give it to Colombia again, take it out of synonymy. And just very quick, I just want to show that in this tree of life, these are two different, they come in two different places. Here is morii and here is Elaphoglossum columbianum. So with that, I want to thank very much. My advisors are NYBG, because I know this is an NYBG event. Barbara Ambrose and Robin Moran, they have always been with me, excited about Elaphoglossum, about diversity of leaves, etc. I also want to thank the students, because I think students are the source of inspiration and energy, and my collaborators in Elaphoglossum, the funding institution, NSF, and my institution, the Fort Worth Botanic Garden, and the Botanical Research Institute of Texas. So thank you so much. And yeah, we'll leave the questions for the end.
Wonderful. Alejandra, thank you so much for that terrific presentation and beautiful photographs of ferns. Once again, we're saving questions at the end, so please submit those to the Q&A if you have not already. Our final speaker today, Dr. Julian Aguirre Santoro, is a botanist and evolutionary biologist who studies the evolution and taxonomy of bromeliads. He's currently an assistant professor and herbarium curator at the National University of Columbia, where he leads several projects on bromeliad conservation and diversity and the evolution of Columbia. Dr. Santoro has also conducted research at the New York Botanical Garden and at the National Museum of Rio de Janeiro, where he carried out botanical expeditions in the Atlantic Forest, the Greater Antilles, in the Caribbean, and in Northwestern South America. So please join me now in welcoming Julian to the stage.
Thank you, Charlie, and good morning, everyone. Okay. So before I start, I want to thank to NYBG, which is an institution I really love. I got my PhD. I spent 5 years of my life there, so it's always good thing to be back in any opportunity. So this time I want to talk about a little bit my research on bromeliads, the pineapple family. And I'm gonna give you a quick overview on how herbarium collections and data repositories, like the ones you are feeding with WeDigBio, are very important for my research. And if not important, they are very critical. All right, before I go deep into why collections are important, I want to introduce bromeliads for those who don't know what they are. Most people know this is the pineapple family, and most people associate bromeliads with these plants that grow as epiphytes and that they have these rosettes that accumulate water and nutrients and debris inside the leaves, which is a very important feature of the bromeliads. But not all of them are like that. So think of the Spanish moss. That's another bromeliad that doesn't have that configuration, but still a bromeliad. What really defines a bromeliad in technical terms are these structures that you can see in the slide, these specialized hairs, you can call them hairs or scales or trichomes. And these specialized trichomes are covering normally the surface of the leaves and other parts of the plants. And what they do is that they allow the plant to absorb water and nutrients from there, so they don't depend exclusively on the roots to take nutrients and water. So this is why bromeliads can grow almost in any surface that you can imagine, on rocks, on trees, on power cords, whatever you want, and also allow them to live in any ecosystem in the tropics. And this very unique adaptation of the bromeliads is what made me very attracted to study this group in depth. And another thing that made me very attracted to study this group is that it's a species-rich group. It has 3,500 species, more or less, and 99% of them are exclusive to the American tropics. So being a South American scientist, that's a huge opportunity for approaching a group that I can understand in depth and discover a lot of important things for my homeland, right, which is the American tropics. Within the American tropics, bromeliads are everywhere in every ecosystem, from deserts to rainforests to high elevation alpine ecosystems. And it is thought that the group appeared for the first time in the Guiana Shield. For those who don't know where the Guiana Shield is these are rocky formations that are in Venezuela, Brazil, and Colombia, and the Guianas. And so they're thought to have originated there. And from there, they dispersed to other parts of the American tropics, like the Andes, the Amazon, the Atlantic forest, the Caribbean. And once they arrived there, well, they diversified locally and they produce a lot of endemism. So that's why this is also a family where you find a lot of species with high importance for conservation. So as you saw from the previous talks, the groups that we choose to study for research, they always have the same attractive features for research. And is that their biodiversity is very important for the region, but it's also very unknown in many aspects. So what attracted me to establish a line of research on this family is that I could study this group from these 3 points of view. One is studying the taxonomy and the discovery of within the group. The second is to study the evolution and biogeography of the group, and third, the conservation. And as you heard from the other talks from Aleja and Matthew, one key to unlock all the mysteries that are behind those lines of research are in the herbarium specimens. So this is why I want to present this slide, and also to show you that herbarium specimens are normally the first thing that we look at when we are approaching a question or a problem on biodiversity. But it's not a linear process. We don't start from the specimen and then always get to a final solution. But sometimes we have to conduct research and look at the data again, and then go back to the herbarium specimens and the repositories to understand the patterns that they're trying to tell us. Okay, so, and this is, herbarium specimens, as you saw, are extremely useful because you can find a lot of information in just one single herbarium sheet. So for example, if you look at the specimen itself, you normally find all the parts, or at least the most important parts of the specimen, which allows you to study in detail the morphology, the shapes, the variation of the structures, and use that information to describe and to look for patterns on the morphological part of the plants. And if you look at the label, well, the label has a ton of information that's normally very useful, especially if the labels are very informative. So I separated the information on the label on three main things that you can get from them. One is you can get a lot of information about the morphology, the biology, the ecology of the plant. You can also find very useful information about where the plant grows, so their geographic location. And you can also tell a story about where these plants were collected and who collected them and when they collected them. So you can not only focus on the biological aspect of it, but also the historical aspect of it. So if this is only present in one specimen, just imagine what you can do with hundreds of them, right? The amount of information that you can get out of them. So now let me give you a quick overview of what I've done in my research, specifically using herbarium specimens. So I normally find ideas for my projects by choosing and studying in detail the taxonomy of genera or lineages that I found very interesting and that I know that are poorly studied, like the ones that you see in this slide. These are groups that I have studied with a lot of detail. I have, to do this, I normally aim to publish a monograph or a synopsis, which is like a full compendium of the information of all the species, and try to give tools to people to learn how to identify them and tell them where they grow, what is their morphological variation. And in that exercise, I have to go and look at the specimens, have to go to the herbarium, look at them with a lot of detail. And because specimens are in many, many herbaria, well, I don't have the money or the time to go to all of them. And that's when databases and images are very helpful for me. So in this process, I get to create a full picture of what the variation in the genus is and what the conservative patterns are, but also the oddities inside the groups. So in the process, it turns out that when you examine specimens, you always identify some odd looking specimens, like Aleja pointed out in her study with the Laphoglossum. And normally those odd species make you get suspicious and at the end conclude that these are new species, right? Just by looking at the morphology. But sometimes when you are looking at the data overall and you put it in as points in a geographic map, you can also identify outliers where the morphology doesn't show a clear-cut pattern that they are new species. But for being outliers, it makes you think again and go look back and look at those specimens. Here's an example of two new species that I described with Fabian Michelangeli. He was my advisor at the New York Botanical Garden. So these two species from the genus Disteganthus, which is an Amazonian genus that is relatively conservative in their morphology variation. The species are hard to tell apart sometimes. You have to look at very detailed structures. But by understanding the patterns of distribution of all the specimens in the whole genus, we were able to identify that these specimens in particular were, especially the three populations that you see in green and the one that you see in orange, were clear outliers from the remaining of the group. So we went back to study in detail those specific three, the specimens from those specific three populations, and realized that what we were looking at were in fact two new species that we call Disteganthus morii and Disteganthus gracie for, well, if you're working in YBG, you know who they are dedicated to. And this same approach, every time I review a genus, I study the taxonomy in depth, like keeps appearing over and over. This is two more examples from my current project with Puya, which these are not epiphytic bromeliads, these are terrestrial ones and from high elevations. And I keep looking at the data from the repositories, like well-curated data, and I keep identifying outliers that then I go back to the herbarium or to the images, look at them and realize that they are in fact new species. Okay, so moving to my second line of research. So the ability to use data from collections does not stop only on taxonomic studies and discovering new species. I use this well-curated information from collections, supported by the good understanding of the groups, to allow me investigate biological patterns, especially evolutionary patterns that explain the origin and the evolution of the plant lineages that I like to study. So this slide shows a summary of what was my doctoral dissertation in New York, where I used DNA to reconstruct the phylogeny of a group of nearly 80 species of a group that I named the Rombergia Alliance. So I'm not gonna explain what a phylogeny is because I think Aleja made it very clear. It's like a genealogical history of the species. It shows who's related with who. But basically what I wanna say here is that with the phylogeny, the relationships that I found as a framework, I was able to look at the data from the collections in a different way. So as you can see in the map, I was able to infer that although the species of this group are mostly distributed near the coast of tropical South America, either in the Pacific part or the Atlantic part, I was able to find out that the species that grow on the Brazilian coast are more closely related to the Caribbean ones, while the Pacific coast ones correspond to a more distant lineage. I was also able to go to herbarium specimens and measure a bunch of flowers from these groups. And I also was able to figure out that within this range of distribution of this group, the flowers in the Caribbean species seem to respond much more rapidly to adaptation than those of the continental part of the distribution of the group, which is a discovery that I made based only on measuring herbarium specimens. All right, and finally, I also use collection data that is well curated for projects on conservation. This is something that I have been involved a lot since I moved back to Colombia to become a professor. And one of the projects that I have been working on is the assessment of extinction risk of endemic bromeliads in Colombia. This image is from a project that I carried out with a lot of colleagues. It's not just me, it's a lot of people working on this with support of the International Union of the Conservation of Nature, the IUCN. So as a specialist in a group of plants like bromeliads, well, I help the IUCN to look at the databases from herbarium specimens to help curate that information and to have a clear picture on the occurrence of endemic species in order to assess the risk of extinction. So this information is very important because it helps lawmakers to make environmental decisions and policies to protect our biodiversity. So here's in this slide is a little example of one of the assessments we did with a Puya exuta, a bromeliad endemic from Colombia, that you can check directly in the REDList website. And this information can also be used locally in combination with the risk assessments to of other groups to inform local authorities on the conservation status of our native flora. The images that you see in this slide are results from the same study on high elevation endemic plants of Colombia, and it's showing how the risk of extinction of endemic species of orchids, bromeliads, and frailejones has changed over the years, and also in what proportion the species are endangered under different categories of risk. And this information, combined with information from collections, also gave us the basis to use a novel approach that is being proposed by the IUCN right now to delimit key biodiversity areas, which are strategic regions that guarantee the protection of highly endangered species with the, let's say, least impact on economic development of the country. This is something that we are working right now with people from the Humboldt Institute and Lady Vallejo, Nelson Salinas, and it will be published very soon. Okay, I want to finish my talk very quickly by saying that collections continue to be my main source of data, and as they become more easily available, my research being here in Colombia, which is relatively remote for where the data is, as it is available, the data, my research is more possible and more impactful. So at the moment, I'm conducting a research project with the genus Puya. This is an Andean lineage of bromeliads, and I'm already starting to look very interesting patterns using databases, creating these databases of collections. Like you can see in this picture where there's a very strong tendency on the phylogeny to follow a latitudinal distribution and also an altitudinal distribution. And also in these hard times of the pandemic, without having access to the lab or to go to the field, collections and databases have been very critical for the research of my students. So with no exception, they are right now compiling information from databases. They're organizing tables and conducting their thesis projects using databases. And this is just a little sample of what the titles of those projects are happening under my guidance. So I finish here. I just want to emphasize that all these projects, I didn't do them on my own. It was always in collaboration with other people. I would like to acknowledge everybody that was involved, but I want to acknowledge especially these people in this picture who have been incredibly supportive in academic life. Thank you very much.
Thank you so much, Julian, and to all our speakers for your talks today. So many great perspectives on different groups of plants from different parts of the world, and we've got great questions coming in, so thank you. And like I said, it's time for the Q&A. So audience members, go ahead and submit your questions now to the Q&A if you haven't yet. And you can also upvote questions that others have already submitted. And this helps us prioritize the questions to get as many as we can today. So let's go over there. Let me make sure that everybody can be seen. All right, so first question from Lori. Has the rate of specimen depositions remained the same over time? And is any change in the deposition rate in herbaria accounted for in the analysis of distribution based on specimens? And this question can be for anyone.
So I'll start off. So that's a really great question. And yes, unfortunately, particularly for temperate areas, collecting of herbarium specimens has really dropped off in the last couple of decades. And the good thing about these data aggregators is not only do they source herbarium specimens, but they also include photographic records from iNaturalist. And so for groups that like orchids that have these cult-like followings of really devoted people who go out every single weekend and photograph every single known population that they know, these iNaturalist records really help supplement current records. And so those iNaturalist records were included in my distribution mapping for the orchids of New York. So, definitely. And I think it's something that all three speakers have stressed, is that there's always a need to collect more specimens, including of rare plants.
Alejandra, did you have to add something?
Yeah, I wanted to add to Matthew that to what Matthew said, that it's true that they are declining, and there are these other great resources as INAT that are kind of like supplying different information. I think permits have been also more difficult. So now for researchers going outside and even like within the US or international, there are more laws, which is sometimes it's very understandable, and I think it's very important for us to collect in a way that it's in line with laws, so that people know that we're collecting for research, but that can be challenging. And so I think that's also has kind of like one of the reasons why this decrease of collections. I want to say that, for example, in the tropics, I do see that iNaturalist is not as used as here, for example. So there are many initiatives that are great, that are kind of like telling the general public and the different communities to use it and to take good pictures. So if you really don't want to collect because you don't have the permits, try to take like great pictures, and those great pictures upload in iNaturalist, we will be able to see them, and we might contact you and do some collecting trips with you if your species are of interest of some research.
Thank you both. Those are great answers. And next up, we have a question about bromeliads. On your map, I think there were bromeliads shown in West Africa. Is that true, Julian? Can you tell us a little bit how they got there?
Yeah, well, that's a mysterious one. That's called Pitcairnea feliciana. It's the only bromeliad that's not in the Americas. Yeah, people have tried to figure out how it got there using DNA data, using phylogenies. This answer is not very satisfactory. Everything indicates that it was a long distance trip that some seed made from Brazil to the West Africa. It's the only plausible answer with the results, but there is no evidence that that could have happened. So we just have to be happy with that answer.
Till we have more data.
Exactly, or better evidence, right? Like fossils or something.
Indeed.
Maybe some digging in West Africa to see if there were other bromeliads there before that one arrived.
Terrific, thank you. Another question, this is coming from Michelle. Thank you all for your wonderful presentations. First of all, I research epiphytes in Panama, and I have a question for Alejandra. Do you know why divided leaves evolved? It sounds like the pinnate species being high in the canopy could be a light or a wind adaptation. Do you have any thoughts?
Yeah, I have had many thoughts over the years about this. They don't let me sleep at night, really, because I always this was the question that we started with, why, and it's very difficult to answer a why question. So that's why we did this. We know that there are 4 species. We did also some evolutionary developmental studies trying to look at the molecular genomics of these differences, which I didn't present. And we have some evidence, but there's just not a clear answer why. I can tell you that the 2 species with the pinnate leaf that I talk about, they likely grow in the canopy, but we need to study them better. As I say, there are not many collections, so maybe it should be just kind of a focus undergrad or even like small project for a researcher that lives close to where these species occur, and kind of survey them and see if they really, if that hypothesis that I have, that they grow only in the canopy, is true. But I also wanted to point out that the other species that have divided leaves, that are not these pinnate leaves, the one that really look like parsley, they don't really grow in the canopy. You can find them lower in the trunks in different cloud forests. So when we were doing this, we kind of plot the different elevations at what the species occur, the simple leaves and the divided leaves ones, and we didn't find any difference. But I do think that there might be something there with the ones that grow in the canopy, and we really don't know. So something to research further. And if you live somewhere close to where the species are, this is something really great that you can do, and you can send me an email and I can help you if you want.
Terrific. Thank you. We have a question from Calais, and I believe this is for Matthew. Do you think the lack of southern orchids filling the gap is because of habitat disappearance or inconsistent temperature fluctuations for the plants, or a combination, or other factors?
Yeah, that's again a really great question, and that's sort of the next step in my study. This was just sort of a pilot study looking at New York because NYBG is based in New York, and I want to expand the project to all the Northeast or potentially the entire east part of the U.S. and possibly expand the focus groups to outside of orchids. Personally, I think it's probably a combination of effects, because both. I would hypothesize that if it was purely climate change driving the retreat of the northern species, then the southern species would expand. But of course, anyone who's been to southern New York knows there's very little undisturbed habitat, and it's things like just development. So building housing subdivisions where a forest used to be, or even a pasture used to be. It's things like heavy metal deposition from pollution in the soil, and that can affect the mycorrhizal fungi that the orchids associate with. So they may not be able to germinate, or they might not be able to acquire the nutrients that they need in order to grow. And there's still very little that we know about soil mycorrhizae. So that's a big open-ended question. And then of course, deer are also a major problem in the Eastern US. I love deer objectively, but they eat everything, and they have a particular taste for orchids. They're succulent leaves, and they're just very palatable. So unfortunately, the overabundance of deer has also been a major issue. And as you hinted at, it's probably a combination of these factors. And to really answer those questions, we have to do additional studies looking at modeling, overlaying different factors like pollution levels onto maps, and trying to understand and tease apart what might be causing that. So definitely more area for research.
Wonderful. Thank you. We have a question from an audience member who does wetlands vegetation research and is interested to learn more about the necessary steps for digitizing collections and making them more accessible. Can those of you who are involved in digitization projects talk about how those get started and how it all happens? Matthew?
Yes. So as I mentioned in my talk, and Charlie mentioned, I lead a multi-institution digitization grant that's funded by the National Science Foundation. And the way we started that is Barbara Tiers, who's the director of the herbarium here at NYBG, and I were talking about, this program that NSF runs is going to run out in a couple of years. We should put in under the grant proposal, what group should we focus on? Partially that's driven by what have we already digitized in our collection, and also what questions we want to ask. So I work on orchids. Of course, as I presented, most of the groups that I personally work on happen to be terrestrial orchids. Orchids are primarily epiphytic. And so we started looking at, sort of the pandas of the plant world, as we like to call them, all the really cool things that the public likes. So orchids, other epiphytes, carnivorous plants, succulents, cacti, and that's how we structured the grant. And the National Science Foundation is very aware that once this particular program runs out, they need to develop a new program that extends the digitization effort. And it may change slightly in focus, but we're very hopeful that it will have continued digitization efforts. Yeah, so it's a little bit of everything, question driven, taxon driven, but there's always more to do. You never finish completely digitizing a collection.
Alejandra, do you have your hand up?
Yes, and I wanted to add to the wetland person. I'm not sure if this was also a question about how to collect and how to make the research more attractive. I think wetlands are amazing for conservation. I think also there are just many other people that work in wetlands in different parts of the world. So I will suggest the person who asked the question to try to get together or just read their papers and try to get together to see how they are also making their research more attractive. And I'm sure there are many plants in wetlands that have specific ways of collecting them. For example, there might be a lot of aquatic plants or algae, and sometimes they are tricky to collect. But if you are interested in that, I will suggest to get in touch with people that are working in other places, also in wetlands. And I'm sure there might be a network, or at least you can start creating one, because collaboration with people that is work in similar habitats can always teach you a lot of things about not only how to research there, but also how to kind of make your research more attractive to the public and to donors.
Thank you so much, Alejandra, for filling out the rest of that question there. We've gone a little bit over time. I hope that's okay with everybody. I put some links in the chat there about our upcoming events, and if you'd like to sign up for anything else. We just have a couple more questions, so if you're willing to stick around, we'll just answer just a couple more, and then we'll close it out for the day. This is a question that was directed towards Matthew, but I think this would be a great question for everybody to answer. Are there areas of digital data that are missing in your particular fields and the groups that you study? And if you had access to the perfect data set for making your research possible, what would that look like? And how would you improve that data as well? And then finally, what can we all do in the future as scientists, herbarium curators, citizen scientists to make that a reality? Julian.
Well, I'm sure Alejandra also knows, but in the country where I live and where we are from, Colombia, it's always like people always talk about our country as the black hole of collections, because there are so many places where people haven't been able to visit to collect plants and see what is in there. So in the case of Colombia, well, we have a deficit on collections. We need more exploration. We need to go to more places if possible. But I also want to say that we have been working on filling that gap really, really hard. I think over the last 20 or 30 years, the amount of botanists working here in Colombia and also other Latin American countries has grown substantially, and we're trying to fill those gaps. Yeah, I think all the data sets could be better. I would love to have many more points in those maps to have a better picture. We all know that whatever is available is not enough for our research. We would like to see more. So yeah, there's always a space for more data. We are waiting for that data, and we as researchers, we don't have the time or the power to do it. So we need help from other people.
Alejandra.
So that's great. Juli, I also think that one of the challenges is to combine that data set. So we here with this FERN digitization grant funded by NSF at the Endless Forum that Matthew has talked about. They are both funded through the same agency, and there is an effort here in the United States to make available the collections through these portals that are called Symbiota, so that you can access many collections in the same place. But I think one of the challenges, and my perfect dreamy data set, will be to have all of these collections in one single place. I know this, like the bioinformatic people might be like, well, that's just crazy. I know it's very difficult, but this is one of, like, my dream will be to do this for the data sets we have, so that we can have all the digitized collections, not only in the US, but also in Colombia, in Latin America, in Asia, in one single place where we can use them all together to see patterns and things like that. A dream.
We got a dream.
I definitely want to second what Alejandra and Julian said, and particularly picking up where Alejandra left off. There's a push within collections to implement what is called the extended specimen network. And again, just as she was talking about, that's really about linking collections, and not only different herbarium collections, but insect collections, fossil collections, soil collections. So, for example, with orchids, if you're trying to understand population declines, you also have to understand the mycorrhizal associations that live in soil. You have to understand the pollinators, so you bring in entomological collections. And that's where I see a lot of research really moving forward, is by leveraging all these different types of collections together to answer some of these really big questions. And in terms of what you can do, you public, at least within the US, by continuing to support the National Science Foundation, call your representative and tell them that basic biological diversity science is important to you, and that supporting museum collections in particular is also really important to you. That's the power of our democracy, and if you make your voice known, that's how we as an institution benefit.
Thank you all for your perspectives on that. And I think those are all really good points and super important. And just to bring this back to citizen science and We Dig Bio, we're all in it together, helping to collect this data as well. So from the very beginning to where the collections are and to making them online, your role is profoundly felt and greatly appreciated. So please keep participating, and if you haven't yet, try out our projects and see if you like them, and you'll find yourself getting sucked in quickly because it's a lot of fun. Thank you so much to our speakers today for joining us, for sharing your perspectives. Thank you so much to our audience for being here today. If you missed any part of this event or wish to watch it again, a recording was made and it will be saved on the NYBG lecture library. We hope many of you will feel inspired by all this to take action and help us to mobilize biodiversity specimens during We Dig Bio and beyond. Check out the links in the chat to register for this afternoon's We Dig Bio Blitz and learn about how to get involved as a virtual volunteer in NYBG's citizen science expeditions. I hope you'll join us for future science, conservation, and humanities lectures from the New York Botanical Garden. On April 16th, we'll learn about the evolutionary development of seed plants as doctoral candidate Cecilia Zumajo shares the results of her PhD research at the New York Botanical Garden. Then on April 22nd, in celebration of Earth Day, we have the premiere of an important new documentary on the conservation of plants, people, and culture on the islands of Vanuatu in the Pacific Archipelago, followed by a conversation with project collaborators from Vanuatu and from the New York Botanical Garden. Please follow the links in the Zoom chat to register for our mailing lists if you haven't already. So thank you all again. Until next time, please stay well, stay green, and take care. Goodbye, everyone.
Thank you. Bye.